So let's begin with mutation. Mutation is a phenomena which results in the alternation of DNA sequences and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomena that leads to the variation in DNA. As you will learn in chapter 5, one DNA helix runs continuously from one end to the other in each chromatid in highly supercoiled form. Therefore, loss means deletion or gain means insertion or duplication of a segment of DNA result in alternation in the chromosomes. Since genes are known to be located on chromosomes, alternation in chromosomes results in the abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arise due to change in a single base pair of DNA. This is known as a point mutation. A classical example of such a mutation is sickle cell anemia. Deletion and insertion of base pairs of DNA causes frame shift mutations. So, in point mutation, only one base pair is changed, and in frame shift mutation, either there is a deletion or there is an insertion of a base pair. There is no deletion or insertion, there is the change of the base pair. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce mutations. These are referred to as mutagens. UV radiation can cause mutations in organisms. It is a mutagen. Now let's see the genetic disorders. First, we need to know what pedigree analysis is. Pedigree analysis. The idea that disorders are inherited has been prevailing in the human society since long. This was based on the heritability of certain characteristics features in families. After the discovery of Mendel's work, the practice of analyzing inheritance pattern of traits in human beings began, since it is evident that control crosses that can be performed in a pea plant or some other organisms are not possible in case of human beings. Study of the family history about inheritance of a particular trait provides an alternative. Such an analysis of traits in a several of generations of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality, or disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure 4.13. As you have studied in this chapter, each and every feature is in any organism is controlled by one or the other gene located on the DNA present in the chromosome. Now, DNA is the carrier of genetic information. It is hence transmitted from one generation to the other without any change or alternation. However, changes or alternations do take place occasionally. Such an alternation or change in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. So, Mendelian disorders. Broadly, genetic disorders may be grouped into two categories: Mendelian disorders and chromosomal disorders. So, Mendelian disorders are like sickle cell anemia, thalassemia, hemophilia, and chromosomal disorders are like Turner syndrome, Down syndrome, Klinefelter syndrome. Mendelian disorders are mainly determined by the alternation or mutation in the single gene. So, alternation or mutation in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principle of inheritance. The pattern of the inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is dominant or recessive. Either the Mendelian disorder is dominant or it is recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait uh, may also be linked to the sex chromosome, as in case of hemophilia. It is evident that this X-linked recessive trait shows transmission from carrier female to male progeny. A representative pedigree is shown in figure 4.14 for dominant and recessive traits. Discuss with your teacher and design pedigrees for characters linked to both autosomes and sex chromosomes. So basically, in डोमिनेंट वाले डिजीज़ में क्या होगा, पेरेंट्स में भी एक डोमिनेंट होगा, एक डिजीज़ होगा, एक पेरेंट डिजीज्ड होगा, होगा ही होगा, और एक प्रोजेनी एटलीस्ट डिजीज्ड होगा. यहाँ पर अगर कोई पेरेंट में भी नहीं है तो यहाँ पे कोई चांस नहीं है कि यहाँ पे हो पाए डिजीज़. यहाँ पर एक पेरेंट है तो यहाँ पर एक चाइल्ड को भी है. एंड इन रेसेसिव वन व्हाट हैपन्स कि पेरेंट में इट मे बी रेसेसिव बिकॉज ऑफ heterozygous condition in parents. The disease may not be visible, but let's suppose that two of the offsprings are getting the homozygous recessive trait gene, so the disease will appear in them. In this also there is no disease in any of the parents, but in offspring there is one diseased child. So now let's talk about color blindness. So color blindness is a sex-linked recessive disorder due to the defect in either red or green cone of eye, resulting in the failure to discriminate between red and green color. This defect is due to the mutation in certain genes present in the X chromosome. It occurs in about 8% of the males and about 0.4% of the females. This is because of the genes that lead to the red-green color blindness are on the X chromosome. Males have only one X chromosome and females have two. The son of a woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. So now let's talk about hemophilia. So this is also a sex-linked recessive disease which shows its transmission from unaffected carrier female to some of the male progeny. Has been widely studied. In this disease, a single protein that is a part of the cascade of proteins involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female is the carrier for hemophilia may transmit the disease to sons. The possibility of female becoming hemophilic is extremely rare because mother of such a female has to be at least a carrier and the father should be hemophilic and viable in the later stage of life. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Now, sickle cell anemia is an autosome linked recessive trait that can be transmitted from parents to the offsprings when both the partners are carrier for the gene or heterozygous. The disease is controlled by a single pair of allele, HBA and HBS. Out of the three possible genotypes, only homozygous individuals for HBS show the diseased phenotype. Heterozygous like HBA and HBS individuals appear apparently unaffected, but they are carrier of the disease, as there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The defect is caused by the substitution of glutamic acid by valine at the sixth position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to the single base substitution at the sixth codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Phenylketonuria we have already done, so it was an inborn error of metabolism is also inherited as the autosomal recessive trait, and in this what was happening, the affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. इस वजह से phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives, and this gets accumulated in the regions of the brain and results in mental retardation. So these are also the excreted through urine because of its poor absorption by kidney. Now, thalassemia is very important from the point of view of questions. Okay. So thalassemia में क्या होता है कि this is also an autosome linked recessive blood disease transmitted from parents to the offsprings when both the parent partners are unaffected carrier for the gene or heterozygous. The defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chains, alpha and beta genes that make up the hemoglobin. This causes the formation of abnormal hemoglobin molecules, resulting into anemia, which is characteristic of this disease. Thalassemia can be classified according to which gene of the hemoglobin molecule is affected. So in alpha thalassemia, production of alpha globin chain is affected, while in beta thalassemia, production of beta globin chain is affected. Alpha thalassemia is controlled by two closely linked genes, HbA1 and HbA2, on chromosome number 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. The more genes affected, the less alpha globin molecules produced. While beta thalassemia is controlled by a single gene, HBB, on chromosome number 11 of each parent, and occurs due to mutation of one or both the genes. So thalassemia differs from sickle cell anemia in that thalassemia is a qualitative problem, and sickle cell anemia, it was a quantitative problem of synthesizing too few globin molecules, while the thalassemia is qualitative problem of synthesizing an incorrectly functioning globin. So this is very important, and from here alpha thalassemia and beta thalassemia में ये चीज़ें यूज़ होती हैं, ये याद रखना है. So now the chromosomal disorders. The chromosomal disorders on the other hand are caused due to the absence or excess or abnormal arrangement of one or more chromosomes. So failure of segregation of chromatids during a cell division cycle results in the gain or loss of a chromosome called aneuploidy. For example, in Down syndrome results in the gain of extra copy of the chromosome 21. Similarly, Turner syndrome results due to the loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomena is known as polyploidy. So this condition is often seen in plants. The total number of chromosomes in a normal human cell is 46, means 23 pairs. So out of these, 22 pairs are autosomes and 1 pair of chromosome are sex chromosome. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual or an individual may lack one of any one pair of chromosomes. So these situations are known as trisomy or monosomy of a chromosome respectively. So such a situation leads to very serious consequences in the individual. So Down syndrome, Turner syndrome, Klinefelter syndrome are the common examples of chromosomal disorders. So let's see what are the symptoms and what happens in all these three diseases. So in Down syndrome, the cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, means trisomy of 21. So this disorder was first described by Langdon Down in 1866. So the affected individual is short-statured with small round head, furrowed tongue, and partially open mouth. Palm is broad with characteristic palm crease, and physical or psychomotor and mental development is retarded. So this is the diagram of the individual affected by Down's syndrome. Now let's see Klinefelter syndrome. In this genetic disorder is also caused due to the presence of an additional copy of X chromosome resulting into karyotype of 47, means XXY. So such an individual has overall masculine development. So however, the feminine development of breast, that is gynecomastia, is also expressed. So such individuals are what? They are sterile. So this is the diagrammatic view. Now coming on to Turner syndrome. Such a disorder is caused due to the absence of one of the X chromosomes, that is 45 with XO. So such females are sterile as ovaries are rudimentary besides other features including lack of other secondary sexual character. So we are done with chapter number 4, that is principles of inheritance and variation.